Hey there, science enthusiasts! Today, we're diving into the fascinating world of fruit flies, also known as Drosophila melanogaster. These tiny creatures are not just your average insect, but they've actually become a key player in the world of genetics and neuroscience. Did you know that fruit flies are a model organism in genetics? That's right, they've been instrumental in understanding the basics of genetics, including inheritance and mutations. Their short lifespan and rapid reproduction make them the perfect subjects for genetic research. But that's not all. Fruit flies are also important in neuroscience and behavioral genetics. They've helped us understand the basics of brain function, memory, and even addiction. So, the next time you see a fruit fly buzzing around, remember that they're not just pesky little insects. They're a valuable tool in the world of science, helping us unlock the mysteries of genetics and neuroscience. Thank you for joining us on this fascinating journey. If you enjoyed this video, please give it a thumbs up, and don't forget to subscribe for more science-filled content. Until next time, keep exploring. Drosophila melanogaster is a species of fly and insect of the order Diptera in the family Drosophilidae. The species is often referred to as the fruit fly or lesser fruit fly, or less commonly the vinegar fly, pomace fly, A5 or banana fly 6. In the wild, D. melanogaster are attracted to rotting fruit and fermenting beverages, and are often found in orchards, kitchens, and pubs. Starting with Charles W. Woodworth's 1901 proposal of the use of this species as a model organism, 7,8 D. melanogaster continues to be widely used for biological research in genetics, physiology, microbial pathogenesis, and life history evolution. D. melanogaster was the first animal to be launched into space in 1947. 9. As of 2017, 6 Nobel Prizes have been awarded to Drosophilists for their work using the insect. 1011 Drosophila melanogaster is typically used in research owing to its rapid life cycle, relatively simple genetics with only 4 pairs of chromosomes, and large number of offspring per generation. 12. It was originally an African species, with all non-African lineages having a common origin. 13. Its geographic range includes all continents, including islands. 14. D. melanogaster is a common pest in homes, restaurants, and other places where food is served. 15. Flies belonging to the family Tephrididae are also called fruit flies. This can cause confusion, especially in the Mediterranean, Australia, and South Africa, where the Mediterranean fruit fly Ceratodus capitata is an economic pest. Etymologist term Drosophila, meaning dew-loving, is a modern scientific Latin adaptation from Greek words delta rho s sigma omicron sigma, drosos, dew, and phi iota lambda alpha, philia, lover. The term melanogaster, meaning black-belly, comes from ancient Greek mulanda alpha sigma, melis, black, and gaster, belly. Physical upper ants female fruit flies are substantially larger than male fruit flies, with females having bodies that are up to 30% larger than an adult male. 1718. Wild-type fruit flies are yellow-brown, with brick-red eyes and transverse black rings across the abdomen. The black portions of the abdomen are the inspiration for the species name melanogaster equals black-bellied. The brick-red color of the eyes of the wild-type fly are due to pigments. 19. Xanthommatin, which is brown and is derived from tryptophan, and drosopterins, which are red and are derived from guanosine triphosphate. 19. They exhibit sexual dimorphism. Females are about 2.5 millimeters, 0.10 in long. Males are slightly smaller. Furthermore, males have a cluster of spiky hairs claspers surrounding the reproducing parts used to attach to the female during mating. Extensive images are found at flybase. 20. Drosophila melanogaster can be distinguished from related species by the following combination of features. Genna approximately 1/10 diameter of eye at greatest vertical height. Wing hyaline and with costal index to 0.4. Male protarsus with a single row of approximately 12 seta forming a sex comb. Male epandrial posterior lobe small and nearly triangular. Female abdominal turgid 6, with dark band running to its ventral margin. Female, ovoscapt small, pale, without dorsodistal depression and with 12 to 13 peg-like outer ovis ancilla. 2120 to Drosophila melanogaster flies can sense air currents with the hairs on their backs. Their eyes are sensitive to slight differences in light intensity and will instinctively fly away when a shadow or other movement is detected. 23. Life cycle and reproduction egg of D. melanogaster under optimal growth conditions at 25 Celsius, 77 degrees Fahrenheit. The D. melanogaster lifespan is about 50 days from egg to death. 24. The developmental period for D. melanogaster varies with temperature, as with many ectothermic species. The shortest development time egg to adult, 7 days, is achieved at 28 Celsius, 80 to F. 25-26 development times increase at higher temperatures, 11 days at 30 C or 86 degrees Fahrenheit due to heat stress. Under ideal conditions, the development time at 25 Celsius, 77 degrees Fahrenheit, is 812 days. 25-26-27 at 18 C 64 F it takes 19 days. 25-26 and at 12 Celsius, 54 degrees Fahrenheit, it takes over 50 days. 25-26 under crowded conditions. Development time increases. 28 while the emerging flies are smaller. 28-29. Females lay some 400 eggs embryos, about 5 at a time, into rotting fruit or other suitable material, such as decaying mushrooms and sap fluxes. Drosophila melanogaster is a holometabolic insect, so it undergoes a full metamorphosis. Their life cycle is broken down into 4 stages: embryo, larva, pupa, adult, 30. The eggs, which are about 0.5 millimeters long, hatch after 12 to 15 hours at 25 C or 77 degrees Fahrenheit. 25-26, the resulting larvae grow for about 4 days at 25 C while molting twice into second and third instar larvae at about 24 and 48 hours after hatching. 25-26, during this time, they feed on the microorganisms that decompose the fruit, as well as on the sugar. The mother puts feces on the egg sacs to establish the same microbial composition in the larvae's guts that has worked positively for herself. 31. Then the larvae encapsulate in the puparium and undergo a 4-day-long metamorphosis at 25 C, after which the adults at close emerge. 25-26 Drosophila melanogaster, commonly known as the fruit fly, has been a significant model organism in embryonic development research. Many of its genes that regulate embryonic development and their mechanisms of action have been crucial in understanding the fundamental principles of embryonic development regulation in many multicellular organisms, including humans. Here are some important genes regulating embryonic development in Drosophila melanogaster and their modes of action. Maternal genes. These genes are encoded in the female fruit fly and are present in the early stages of embryo development. They determine the embryo's main features and early development. For example, the gene called becoid regulates the formation of the embryo's anterior end, and its absence leads to an embryo lacking a head. Zygotic genes. These genes are activated in later stages of embryo development when the fruit fly embryo begins to produce its own genetic products. For example, the hunchback gene regulates the formation of segments in the embryo. Citation needed. Homeotic genes. This gene family regulates segmentation and axial patterning in development. They act as regulatory factors that determine cell fate in embryonic development. For example, the gene called antennapedia regulates the formation of anterior limbs in the embryo. Morphogens. These are molecules that form gradients in embryonic development and regulate cell fate depending on their position in the gradient. For example, the hedgehog morphogen regulates the differentiation of segments and segment identity in the fruit fly embryo. These genes and their modes of action form a complex regulatory network that guides the embryonic development of Drosophila melanogaster. They influence cell differentiation, segment formation, and axial patterning in the embryo, ultimately leading to the development of a fully formed adult fruit fly. Duration: 35 seconds. 0:35 sound of Drosophila heartbeat moles perform a sequence of 5 behavioral patterns to court females. First, males orient themselves while playing a courtship song by horizontally extending and vibrating their wings. Soon after, the male positions himself at the rear of the female's abdomen in a low posture to tap and lick the female genitalia. Finally, the male curls his abdomen and attempts copulation. Females can reject males by moving away, kicking, and extruding their ovipositor. 30 to copulation lasts around 15 to 20 minutes, 33 during which males transfer a few hundred, very long 1.76 millimeters, sperm cells and seminal fluid to the female. 34. Females store the sperm in a tubular receptacle and in 2 mushroom-shaped spermathecae. Sperm from multiple matings compete for fertilization. A last male precedence is believed to exist. The last male to mate with a female sires about 80% of her offspring. This precedence was found to occur through both displacement and incapacitation. 35. The displacement is attributed to sperm handling by the female fly as multiple matings are conducted and is most significant during the first 1 to 2 days after copulation. Displacement from the seminal receptacle is more significant than displacement from the spermathecae. 35. Incapacitation of first male sperm by second male sperm becomes significant 2 to 7 days after copulation. The seminal fluid of the second male is believed to be responsible for this incapacitation mechanism without removal of first male sperm, which takes effect before fertilization occurs. 35. The delay in effectiveness of the incapacitation mechanism is believed to be a protective mechanism that prevents a male fly from incapacitating his own sperm should he mate with the same female fly repetitively. Sensory neurons in the uterus of female D. melanogaster respond to a male protein, sex peptide, which is found in semen. 36. This protein makes the female reluctant to copulate for about 10 days after insemination. The signal pathway leading to this change in behavior has been determined. The signal is sent to a brain region that is a homologue of the hypothalamus, and the hypothalamus then controls sexual behavior and desire. 36. Gonadotropic hormones in Drosophila maintain homeostasis and govern reproductive output via cyclic interrelationship, not unlike the mammalian estrous cycle. 37. Sex peptide perturbs this homeostasis and dramatically shifts the endocrine state of the female by inciting juvenile hormone synthesis in the corpus allodum. 38. D. melanogaster is often used for life extension studies, such as to identify genes purported to increase lifespan when mutated. 39. D. melanogaster is also used in studies of aging. Werner syndrome is a condition in humans characterized by accelerated aging. It is caused by mutations in the gene WRN that encodes a protein with essential roles in repair of DNA damage. Mutations in the D. melanogaster homologue of WRN also cause increased physiologic signs of aging, such as shorter lifespan, higher tumor incidence, muscle degeneration, reduced climbing ability, altered behavior, and reduced locomotor activity. 40. Myosince-meiotic recombination in D. melanogaster appears to be employed in repairing damage in germline DNA as indicated by the findings that meiotic recombination is induced by the DNA-damaging agents ultraviolet light. 41. And mitomycin C40 to DNA repair is a collection of processes by which a cell identifies and corrects damage to the DNA molecules that encode its genome. 1. In human cells, both normal metabolic activities and environmental factors such as radiation can cause DNA damage, resulting in tens of thousands of individual molecular lesions per cell per day. 2. Many of these lesions cause structural damage to the DNA molecule and can alter or eliminate the cell's ability to transcribe the gene that the affected DNA encodes. Other lesions induce potentially harmful mutations in the cell's genome, which affect the survival of its daughter cells after it undergoes mitosis. As a consequence, the DNA repair process is constantly active as it responds to damage in the DNA structure. When normal repair processes fail, and when cellular apoptosis does not occur, irreparable DNA damage may occur. This can eventually lead to malignant tumors or cancer as per the two-hit hypothesis. The rate of DNA repair depends on various factors, including the cell type, the age of the cell, and the extracellular environment. A cell that has accumulated a large amount of DNA damage or can no longer effectively repair its DNA may enter one of three possible states. Natural DNA damage is an alteration in the chemical structure of DNA, such as a break in a strand of DNA, a nucleobase missing from the backbone of DNA, or a chemically changed base, such as 8-OHDG. DNA damage can occur naturally or via environmental factors, but is distinctly different from mutation, although both are types of error in DNA. DNA damage is an abnormal chemical structure in DNA, while a mutation is a change in the sequence of base pairs. DNA damages cause changes in the structure of the genetic material and prevents the replication mechanism from functioning and performing properly. 1. The DNA damage response (DDR) is a complex signal transduction pathway which recognizes when DNA is damaged and initiates the cellular response to the damage. DNA damage and mutation have different biological consequences. While most DNA damages can undergo DNA repair, such repair is not 100% efficient. Unrepaired DNA damages accumulate in non-replicating cells, such as cells in the brains or muscles of adult mammals, and can cause aging. 3. For 5, also see DNA damage theory of aging. In replicating cells, such as cells lining the colon, errors occur upon replication of past damages in the template strand of DNA or during repair of DNA damages. These errors can give rise to mutations or epigenetic alterations. 6. Both of these types of alteration can be replicated and passed on to subsequent cell generations. These alterations can change gene function or regulation of gene expression and possibly contribute to progression to cancer. Throughout the cell cycle, there are various checkpoints to ensure the cell is in good condition to progress to mitosis. The three main checkpoints are at G1 per second, G2M, and at the spindle assembly checkpoint regulating progression through anaphase. G1 and G2 checkpoints involve scanning for damaged DNA. 7. During S phase, the cell is more vulnerable to DNA damage than any other part of the cell cycle. G2 checkpoint checks for damaged DNA and DNA replication completeness. Medical Usicit is given intravenously to treat upper gastrointestinal cancers, e.g. esophageal carcinoma, anal cancers, and breast cancers, as well as by bladder instillation for superficial bladder tumors. Mitomycin C has also been used topically rather than intravenously in several areas. The first is cancers, particularly bladder cancers and intraperitoneal tumors. It is now well known that a single instillation of this agent within 6 hours of bladder tumor resection can prevent recurrence. The second is in eye surgery, where mitomycin C 0.02% is applied topically to prevent scarring during glaucoma filtering surgery and to prevent haze after PRK or LASIK. Mitomycin C has also been shown to reduce fibrosis in strabismus surgery. 6. The third is in esophageal and tracheal stenosis, where application of mitomycin C onto the mucosa immediately following dilatation will decrease restenosis by decreasing the production of fibroblasts and scar tissue. In April 2020, mitomycin gel, sold under the brand name Gelmito, was approved in the United States for the treatment of low-grade upper tract urothelial cancer UTUC. 789 urothelial cancer is a cancer of the lining of the urinary system. 7. Mitomycin is also used as a chemotherapeutic agent in glaucoma surgery. Contraindication: tranter women should not take mitomycin gel because it may cause harm to a developing fetus or newborn baby. 7. Females become receptive to courting males about 8 to 12 hours after emergence. 43 specific neuron groups in females have been found to affect copulation behavior and mate choice. One such group in the abdominal nerve cord allows the female fly to pause her body movements to copulate. 36. Activation of these neurons induces the female to cease movement and orient herself towards the male to allow for mounting. If the group is inactivated, the female remains in motion and does not copulate. Various chemical signals, such as male pheromones, often are able to activate the group. 36. Also, females exhibit mate choice copying. When virgin females are shown other females copulating with a certain type of male, they tend to copulate more with this type of male afterwards than naive females, which have not observed the copulati on of others. This behavior is sensitive to environmental conditions, and females copulate less in bad weather conditions. 44. Unless this section needs additional citations for verification, please help improve this article by adding citations to reliable sources in this section. Unsourced material may be challenged and removed. October 2015. Learn how and when to remove this message. Duration: 1 minute and 36 seconds. 136. Courtship behavior in male. The male first showed wing extension stage 1, and later other steps, such as abdomen bending, then frequent attempted copulation, licking, and even ejaculation stage 3. Finally, the male fell over and was on its back. Melanogaster males exhibit a strong reproductive learning curve. That is, with sexual experience, these flies tend to modify their future mating behavior in multiple ways. These changes include increased selectivity for courting only intraspecifically, as well as decreased courtship times. Sexually naive D-melanogaster males are known to spend significant time courting interspecifically, such as with D-simulans flies. Naive D-melanogaster will also attempt to court females that are not yet sexually mature, and other males. D-melanogaster males show little to no preference for D-melanogaster females over females of other species or even other male flies. However, after D-simulans or other flies incapable of copulation have rejected the male's advances, D-melanogaster males are much less likely to spend time courting nonspecifically in the future. This apparent learned behavior modification seems to be evolutionarily significant, as it allows the males to avoid investing energy into futile sexual encounters. 45. In addition, males with previous sexual experience modify their courtship dance when attempting to mate with new females. The experienced males spend less time courting, so have lower mating latencies, meaning that they are able to reproduce more quickly. This decreased mating latency leads to a greater mating efficiency for experienced males over naive males. 46. This modification also appears to have obvious evolutionary advantages, as increased mating efficiency is extremely important in the eyes of natural selection. Polygamy. Both male and female D-melanogaster flies act polygamously, having multiple sexual partners at the same time. 47. In both males and females, polygamy results in a decrease in evening activity compared to virgin flies, more so in males than females. 47. Evening activity consists of those in which the flies participate other than mating and finding partners, such as finding food. 48. The reproductive success of males and females varies, because a female only needs to mate once to reach maximum fertility. 48. Mating with multiple partners provides no advantage over mating with one partner, so females exhibit no difference in evening activity between polygamous and monogamous individuals. 48. For males, however, mating with multiple partners increases their reproductive success by increasing the genetic diversity of their offspring. 48. This benefit of genetic diversity is an evolutionary advantage because it increases the chance that some of the offspring will have traits that increase their fitness in their environment. The difference in evening activity between polygamous and monogamous male flies can be explained with courtship. For polygamous flies, Their reproductive success increases by having offspring with multiple partners, and therefore they spend more time and energy on courting multiple females. 48. On the other hand, monogamous flies only court one female and expend less energy doing so. 48. While it requires more energy for male flies to court multiple females, the overall reproductive benefits it produces has kept polygamy as the preferred sexual choice. 48. The mechanism that affects courtship behavior in Drosophila is controlled by the oscillator neurons DN1S and LNDs. 49. Oscillation of the DN1 neurons was found to be affected by sociosexual interactions and is connected to mating-related decrease of evening activity. 49. Model organism in genetics, melanogaster remains one of the most studied organisms in biological research, particularly in genetics and developmental biology. It is also employed in studies of environmental mutagenesis. History of use in genetic analysis. Alfred Sturtevant's Drosophila melanogaster genetic linkage map. This was the first successful gene mapping work and provides important evidence for the chromosome Th. Theory of inheritance. The map shows the relative positions of allelic characteristics on the second Drosophila chromosome. The distance between the gene's map units are equal to the percentage of crossing over events that occurs between different alleles. D. Melanogaster was among the first organisms used for genetic analysis, and today it is one of the most widely used and genetically best known of all eukaryotic organisms. All organisms use common genetic systems. Therefore, comprehending processes such as transcription and replication in fruit flies helps in understanding these processes in other eukaryotes, including humans. 50. Thomas Hunt Morgan began using fruit flies in experimental studies of heredity at Columbia University in 1910 in a laboratory known as the fly room. The fly room was cramped with 8 desks, each occupied by students and their experiments. They started off experiments using milk bottles to rear the fruit flies and handheld lenses for observing their traits. The lenses were later replaced by microscopes, which enhanced their observations. Morgan and his students eventually elucidated many basic principles of heredity, including sex-linked inheritance, epistasis, multiple alleles, and gene mapping. 50D. Melanogaster had historically been used in laboratories to study genetics and patterns of inheritance. However, D. melanogaster also has importance in environmental mutagenesis research, allowing researchers to study the effects of specific environmental mutagens. 51. Reasons for use in laboratories. Melanogaster multiple mutants, clockwise from top. Brown eyes and black cuticle to mutations. Cinnabar eyes and wild-type cuticle 1 mutation. Sepia eyes and ebony cuticle. Vermilion eyes and yellow cuticle. White eyes and yellow cuticle. Wild-type eyes and yellow cuticle. There are many reasons the fruit fly is a popular choice as a model organism. Citation needed. Its care and culture require little equipment, space, and expense even when using large cultures. It can be safely and readily anesthetized, usually with ether, carbon dioxide gas, by cooling, or with products such as FlyNap. Its morphology is easy to identify once anesthetized. It has a short generation time, about 10 days at room temperature, so several generations can be studied within a few weeks. It has a high fecundity. Females lay up to 100 eggs per day, and perhaps 2,000 in a lifetime. 12. Males and females are readily distinguished, and virgin females can be easily identified by their light-colored, translucent abdomen, facilitating genetic crossing. The mature larva has giant chromosomes in the salivary glands called polytene chromosomes, puffs, which indicate regions of transcription, hence gene activity. The underreplication of rDNA occurs, resulting in only 20% of DNA compared to the brain. Compared to the 47% less rDNA in Sarcophaga barbata ovaries. It is only for pairs of chromosomes, 3 autosomes, and 1 pair of sex chromosomes. Males do not show meiotic recombination, facilitating genetic studies. Recessive lethal balancer chromosomes, carrying visible genetic markers, can be used to keep stocks of lethal alleles in a heterozygous state, without recombination due to multiple inversions in the balancer. The development of this organism from fertilized egg to mature adult is well understood. Genetic transformation techniques have been available since 1987. One approach of inserting foreign genes into the Drosophila genome involves p-elements. The transposable p-elements, also known as transposons, are segments of bacterial DNA that are transferred into the fly genome. Transgenic flies have already contributed to many scientific advances, e.g., modeling such human diseases as Parkinson's, neoplasia, obesity, and diabetes. Its complete genome was sequenced and first published in 2000. 50 sexual mosaics can be readily produced, providing an additional tool for studying the development and behavior of these flies. 53. Genetic markers. See also abdominal pigmentation in Drosophila melanogaster. Melanogaster, which carries the psi allele right, hence showing a characteristic phenotype of curly wings in adult flies. 50 for genetic markers are commonly used in Drosophila research, for example within balancer chromosomes or p-element inserts, and most phenotypes are easily identifiable either with the naked eye or under a microscope. In the list of a few common markers below, the allele symbol is followed by the name of the gene affected and a description of its phenotype. Note, recessive alleles are in lowercase, while dominant alleles are capitalized. Psi1, curly, the wings curve away from the body, flight may be somewhat impaired. 1. Ebony, black body and wings. Heterozygotes are also visibly darker than wild-type SB1. Stubble, bristles are shorter and thicker than wild-type U1. White, eyes lack pigmentation and appear white. Brown, eye color determined by various pigments combined. Y1, yellow, body pigmentation and wings appear yellow, the fly analog of albinism. Classic genetic mutations. Drosophil genes are traditionally named after the phenotype they cause when mutated. For example, the absence of a particular gene in Drosophila will result in a mutant embryo that does not develop a heart. Scientists have thus called this gene Tin Man, named after the Oz character of the same name. 55. Likewise, changes in the shaven baby gene cause the loss of dorsal cuticular hairs in Drosophila sec-Elia larvae. 56. This system of nomenclature results in a wider range of gene names than in other organisms. B. Black. The black mutation was discovered in 1910 by Thomas Hunt Morgan. 57. The black mutation results in a darker colored body, wings, veins, and segments of the fruit fly's leg. 58. This occurs due to the fly's inability to create beta-alanine, a beta amino acid. 57. The phenotypic expression of this mutation varies based on the genotype of the individual. For example, whether the specimen is homozygotic or heterozygotic results in a darker or less dark appearance. 5. 8. This genetic mutation is X-linked recessive. 59 BW. Brown. The brown eye mutation results from inability to produce or synthesize pteridine red pigments due to a point mutation on chromosome 2. 60 meters. Miniature. One of the first records of the miniature mutation of wings was also made by Thomas Hunt Morgan in 1911. He described the wings as having a similar shape as the wild-type phenotype. However, their miniature designation refers to the lengths of their wings, which do not stretch beyond their body and, thus, are notably shorter than the wild-type length. Miniature wings are 1.5x shorter than wild-type, but are believed to have the same number of cells. This is due to the lack of complete flattening by these cells, making the overall structure of the wing seem shorter in comparison. The pathway of wing expansion is regulated by a signal receptor pathway, where the neural hormone bursicon interacts with its complementary G protein-coupled receptor. This receptor drives one of the G protein subunits to signal further enzyme activity and results in development in the wing, such as apoptosis and growth. 63 say. Sepia. The eye color of the sepia mutant is sepia, a reddish-brown color. In wild-type flies, omochromes brown and drosopterins red give the eyes the typical red color. 60 for 65. The drosopterins are made via a pathway that involves a pyrimidinoidin synthase. 66, which is encoded on chromosome 3L. The gene has a premature stop codon in sepia flies, so that the flies cannot produce the pyrimidinoidin synthase and thus no red pigment, so that the eyes stay sepia. 60 for the sepia allele is recessive and thus offspring from sepia flies and homozygous wild-type flies has red eyes. The sepia phenotype does not depend on the sex of the fly. 67V. Vermilion. The vermilion mutants cannot produce the brown omochromes, leaving the red drosopterins, so that the eyes are vermilion-colored, a radiant red, compared to a wild-type D. melanogaster. The vermilion mutation is sex-linked and recessive. The gene that is defect lies on the X chromosome. 68. The brown omochromes are synthesized from kynurenine, which is made from tryptophan. Vermilion flies cannot convert tryptophan into kynurenine and thus cannot make omochromes either. 68. Vermilion mutants live longer than wild-type flies. This longer lifespan may be associated with the reduced amount of tryptophan converted to kynurenine in vermilion flies. 69. Triple mutant male fruit fly Drosophila melanogaster exhibiting black body, vestigial wings, and brown eyes mutations in vestigial, a spontaneous mutation discovered in 1919 by Thomas Morgan and Calvin Bridges. Vestigial wings are those not fully developed and that have lost function. Since the discovery of the vestigial gene in Drosophila melanogaster, there have been many discoveries of the vestigial gene in other vertebrates and their functions within the vertebrates. 70. The vestigial gene is considered to be one of the most important genes for wing formation, but when it becomes overexpressed, the issue of ectopic wings begin to form. 71. The vestigial gene acts to regulate the expression of the wing imaginal discs in the embryo, and acts with other genes to regulate the development of the wings. A mutated vestigial allele removes an essential sequence of the DNA required for correct development of the wings. 72W. White. Drosophila melanogaster wild type typically expresses a brick red eye color. The white eye mutation in fruit flies is caused due to the absence of two pigments associated with red and brown eye colors, pyridines red and omochromes brown. 65. In January 1910, Thomas Hunt Morgan first discovered the white gene and denoted it as W. The discovery of the white eye mutation by Morgan brought about the beginnings of genetic experimentation and analysis of Drosophila melanogaster. Hunt eventually discovered that the gene followed a similar pattern of inheritance related to the meiotic segregation of the X chromosome. He discovered that the gene was located on the X chromosome with this information. This led to the discovery of sex-linked genes and also to the discovery of other mutations in Drosophila melanogaster. 73. The white eye mutation leads to several disadvantages in flies, such as a reduced climbing ability, shortened lifespan, and lowered resistance to stress when compared to wild-type flies. 70 for Drosophila melanogaster has a series of mating behaviors that enable them to copulate within a given environment and therefore contribute to their fitness. The yellow gene is a genetic mutation known as DMEL, Y within the widely used database called Flybase. This mutation can be easily identified by the atypical yellow pigment observed in the cuticle of the adult flies and the mouth pieces of the larva. 76. The Y mutation comprises the following phenotypic classes: the mutants that show a complete loss of pigmentation from the cuticle Y type, and other mutants that show a mosaic pigment pattern, with some regions of the cuticle wild type, Y2 type. 77. Genomegenomic information launchers chromosomes to scale with megabase spare references oriented as in the National Center for Biotechnology Information Database. Cindy Morgan distances are approximate and estimated from the locations of selected mapped loci. The genome of D. melanogaster sequenced in 2000 and curated at the Flybase database 50 to contains for pairs of chromosomes, an X, Y pair, and 3 autosomes labeled to 3, and 4. The fourth chromosome is relatively very small and therefore often ignored, aside from its important eyeless gene. The D. melanogaster sequence genome of 139.5 million base pairs has been annotated 79 and contains around 15,682 genes, according to Ensemble Release 73. More than 60% of the genome appears to be functional non-protein coding DNA 80 involved in gene expression control. Determination of sex in Drosophila occurs by the X, a ratio of X chromosomes to autosomes, not because of the presence of a Y chromosome, as in human sex determination. Although the Y chromosome is entirely heterochromatic, it contains at least 16 genes, many of which are thought to have male-related functions. 81 There are 3 transferrin orthologs, all of which are dramatically divergent from those known in chordate models. 82. Similarity to Umansa June 2001 study by National Human Genome Research Institute comparing the fruit fly and human genome estimated that about 60% of genes are conserved between the two species. 83 About 75% of known human disease genes have a recognizable match in the genome of fruit flies. 84 and 50% of fly protein sequences have mammalian homologs. Citation needed. An online database called Homophila is available to search for human disease gene homologs in flies and vice versa. 85 Drosophila is being used as a genetic model for several human diseases, including the neurodegenerative disorders Parkinson's, Huntington's, spinocerebellar ataxia, and Alzheimer's disease. 86. The fly is also being used to study mechanisms underlying aging and oxidative stress, immunity, diabetes, and cancer, as well as drug abuse. Development Mine article, Drosophila embryogenesis life cycle of this insect has 4 stages: fertilized egg, larva, pupa, and adult. 14 Embryogenesis in Drosophila has been extensively studied as its small size, short generation time, and large brood size makes it ideal for genetic studies. It is also unique among model organisms in that cleavage occurs in a syncytium. Melanogaster oogenesis. During oogenesis, cytoplasmic bridges called ring canals connect the forming oocyte to nurse cells. Nutrients and developmental control molecules move from the nurse cells into the oocyte. In the figure to the left, the forming oocyte can be seen to be covered by follicular support cells. After fertilization of the oocyte, the early embryo, or syncytial embryo, undergoes rapid DNA replication and 13 nuclear divisions until about 5,000 to 6,000 nuclei accumulate in the unseparated cytoplasm of the embryo. By the end of the 8th division, most nuclei have migrated to the surface, surrounding the yolk sac, leaving behind only a few nuclei, which will become the yolk nuclei. After the 10th division, the pole cells form at the posterior end of the embryo, segregating the germline from the syncytium. Finally, after the 13th division, cell membranes slowly invaginate, dividing the syncytium into individual somatic cells. Once this process is completed, gastrulation starts. 90 nuclear division in the early Drosophila embryo happens so quickly, no proper checkpoints exist, so mistakes may be made in division of the DNA. To get around this problem, the nuclei that have made a mistake detach from their centrosomes and fall into the center of the embryo yolk sac, which will not form part of the fly. The gene network transcriptional and protein interactions governing the early development of the fruit fly embryo is one of the best understood gene networks to date, especially the patterning along the anteroposterior AP and dorsoventral DV axes. See under. Morphogenesis. 90 The embryo undergoes well-characterized morphogenetic movements during gastrulation and early development, including germ band extension, formation of several furrows, ventral invagination of the mesoderm, and posterior and anterior invagination of endoderm gut, as well as extensive body segmentation until finally hatching from the surrounding cuticle into a first instar larva. During larval development, tissues known as imaginal discs grow inside the larva. Imaginal discs develop to form most structures of the adult body, such as the head, legs, wings, thorax, and genitalia. Cells of the imaginal discs are set aside during embryogenesis and continue to grow and divide during the larval stages, unlike most other cells of the larva, which have differentiated to perform specialized functions and grow without further cell division. At metamorphosis, the larva forms a pupa, inside which the larval tissues are reabsorbed and the imaginal tissues undergo extensive morphogenetic movements to form adult structures. Immunotype D melanogaster immune system can be divided into two responses, humoral and cell-mediated. Other pathways including the stress response pathways YXSTAT and P38, nutritional signaling via FOXO, and JNK cell death signaling are all involved in key physiological responses to infection. The fat body is the primary secretory organ and produces key immune molecules upon infection, such as serine proteases and antimicrobial peptides AMPs. AMPs are secreted into the hemolymph and bind infectious bacteria and fungi, killing them by forming pores in their cell walls or inhibiting intracellular processes. The cellular immune response instead refers to the direct activity of blood cells, hemocytes, and Drosophila, which are analogous to mammalian monocytes, macrophages. Hemocytes also possess a significant role in mediating humoral immune responses, such as the melanization reaction. 110 The immune response to infection can involve up to 2,423 genes, or 13.7% of the genome. Although the fly's transcriptional response to microbial challenge is highly specific to individual pathogens, Drosophila differentially expresses a core group of 252 genes upon infection with most bacteria. This core group of genes is associated with gene ontology categories such as antimicrobial response, stress response, secretion, neuron-like, reproduction, and metabolism, among others. 111-112 Drosophila also possesses several immune mechanisms to both shape the microbiota and prevent excessive immune responses upon detection of microbial stimuli. For instance, secreted PGRPs with amidase activity scavenge and degrade immunostimulatory dup-type PGN in order to block IMD activation. 113. Unlike mammals, Drosophila have innate immunity but lack an adaptive immune response. However, the core elements of this innate immune response are conserved between humans and fruit flies. As a result, the fruit fly offers a useful model of innate immunity for disentangling genetic interactions of signaling and effector function, as flies do not have to contend with interference of adaptive immune mechanisms that could confuse results. Various genetic tools, protocols, and assays make Drosophila a classical model for studying the innate immune system. 114, which has even included immune research on the International Space Station. 115. YXSTAT signaling. Multiple elements of the Drosophila YXSTAT signaling pathway bear direct homology to human YXSTAT pathway genes. YXSTAT signaling is induced upon various organismal stresses, such as heat stress, dehydration, or infection. The mechanisms through which many of these proteins act is still under investigation. For instance, the TEPs appear to promote phagocytosis of gram-positive bacteria and the induction of the toll pathway. As a consequence, flies lacking TEPs are susceptible to infection by toll pathway challenges. 116. The cellular response to infection. Circulating hemocytes are key regulators of infection. This has been demonstrated both through genetic tools to generate flies lacking hemocytes, or through injecting microglass beads or lipid droplets that saturate hemocyte ability to phagocytose a secondary infection. 119. 120 flies treated like this fail to phagocytose bacteria upon infection and are correspondingly susceptible to infection. 121. These hemocytes derive from two waves of hematopoiesis, one occurring in the early embryo and one occurring during development from larva to adult. 122. However, Drosophila hemocytes do not renew over the adult lifespan, and so the fly has a finite number of hemocytes that decrease over the course of its lifespan. 123 hemocytes are also involved in regulating cell cycle events and apoptosis of aberrant tissue, e.g. cancerous cells, by producing IgG, a tumor necrosis factor signaling molecule that promotes JNK signaling and ultimately cell death, and apoptosis. 124. Behavioral Genetics and Neuroscience, 1971. Ron Konopka and Seymour Benzer published clock mutants of Drosophila melanogaster, a paper describing the first mutations that affected an animal's behavior. IOR. Wild-type flies show an activity rhythm with a frequency of about a day 24 hours. They found mutants with faster and slower rhythms, as well as broken rhythms flies that move and rest in random spurts. Work over the following 30 years has shown that these mutations and others like them affect a group of genes and their products that form a biochemical or biological clock. This clock is found in a wide range of fly cells, but the clock-bearing cells that control activity are several dozen neurons in the fly's central brain. Citation needed. Since then, Benzer and others have used behavioral screens to isolate genes involved in vision, olfaction, audition, learning, memory, courtship, pain, and other processes, such as longevity. Citation needed. Following the pioneering work of Alfred Henry Sturtevant 125 and others, Benzer and colleagues 53 used sexual mosaics to develop a novel fate mapping technique. This technique made it possible to assign a particular characteristic to a specific anatomical location. For example, this technique showed that male courtship behavior is controlled by the brain. 53 mosaic fate mapping also provided the first indication of the existence of pheromones in this species. 126. Males distinguish between conspecific males and females and direct persistent courtship preferentially toward females thanks to a female-specific sex pheromone, which is mostly produced by the female's turgets. The first learning and memory mutants, duns, rutabaga, etc., were isolated by William Chip Quinn while in Benzer's lab, and were eventually shown to encode components of an intracellular signaling pathway involving cyclic AMP, protein kinase, and a transcription factor known as CREB. These molecules were shown to be also involved in saptic plasticity in Aplysia and mammals. 127. The Nobel Prize in Physiology or Medicine for 2017 was awarded to Jeffrey C. Hall, Michael Rosbash, Michael W. Young for their works using fruit flies and understanding the molecular mechanisms controlling the circadian rhythm. 128. Male flies sing to the females during courtship using their wings to generate sound, and some of the genetics of sexual behavior have been characterized. In particular, the fruitless gene has several different splice forms, and male flies expressing female splice forms have female-like behavior and vice versa. The TRP channels NOMP, Nanchun, and inactive are expressed in sound-sensitive Johnston's organ neurons and participate in the transduction of sound. 129. 130. Mutating the genderblend gene, also known as CG6070, alters the sexual behavior of Drosophila, turning the flies bisexual. 131. Flies use a modified version of bloom filters to detect novelty of odors, with additional features including similarity of novel odor to that of previously experienced examples, and time elapsed since previous experience of the same odor. 132. Aggression. As with most insects, aggressive behaviors between male flies commonly occur in the presence of courting a female and when competing for resources. Such behaviors often involve raising wings and legs towards the opponent and attacking with the whole body. 133. Thus, it often causes wing damage, which reduces their fitness by removing their ability to fly and mate. 130. For acoustic communication, order for aggression to occur, male flies produce sounds to communicate their intent. A 2017 study found that songs promoting aggression contain pulses occurring at longer intervals. 135 RNA sequencing from fly mutants displaying over-aggressive behaviors found more than 50 auditory-related genes important for transient receptor potentials, Ca2+ signaling, and mechanoreceptor potentials to be upregulated in the ab neurons located in Johnston's organ. 135. In addition, aggression levels were reduced when these genes were knocked out via RNA interference. 135. This signifies the major role of hearing as a sensory modality in communicating aggression. Pheromone signaling other than hearing. Another sensory modality that regulates aggression is pheromone signaling, which operates through either the olfactory system or the gustatory system, depending on the pheromone. 136. An example is CVA, an anti-aphrodisiac pheromone used by males to mark females after copulation and to deter other males from mating. 137. This male-specific pheromone causes an increase in male-male aggression when detected by another male's gustatory system. 136. However, upon inserting a mutation that makes the flies irresponsive to CVA, no aggressive behaviors were seen. 138. Competition for food specifically. When competing for food, aggression occurs based on amount of food available and is independent of any social interactions between males. 139. Specifically, sucrose was found to stimulate gustatory receptor neurons, which was necessary to stimulate aggression. 139. However, once the amount of food becomes greater than a certain amount, the competition between males lowers. 139. This is possibly due to an overabundance of food resources. On a larger scale, food was found to determine the boundaries of a territory since flies were observed to be more aggressive at the food's physical perimeter. Effective sleep deprivation, however, like most behaviors requiring arousal and wakefulness, aggression was found to be impaired via sleep deprivation. Specifically, this occurs through the impairment of octopamine and dopamine signaling, which are important pathways for regulating arousal in insects. 140. 141. Due to reduced aggression, sleep-deprived male flies were found to be disadvantaged at mating compared to normal flies. 141. However, when octopamine agonists were administered upon these sleep-deprived flies, aggression levels were seen to be increased and sexual fitness was subsequently restored. 141. Therefore, this finding implicates the importance of sleep in aggression between male flies. Vision stereo images of the eye compound eye of the fruit fly contains 760 unit eyes or ommatidia, and are one of the most advanced among insects. Each ommatidium contains 8 photoreceptor cells, R1 to 8, support cells, pigment cells, and a cornea. Wild-type flies have reddish pigment cells, which serve to absorb excess blue light, so the fly is not blinded by ambient light. Eye color genes regulate cellular vesicular transport. The enzymes needed for pigment synthesis are then transported to the cell's pigment granule, which holds pigment precursor molecules. 65 frontal view. Each photoreceptor cell consists of 2 main sections, the cell body and the rhabdomere. The cell body contains the nucleus, while the 100 long rhabdomere is made up of toothbrush-like stacks of membrane called microvilli. Each microvillus is 1 to 2 micrometers in length and about 60 nanometers in diameter. 142. The membrane of the rhabdomere is packed with about 100 million opsin molecules, the visual protein that absorbs light. The other visual proteins are also tightly packed into the microvilli, leaving little room for cytoplasm. Opsins and spectral sensitivity arrangement of the photoreceptor cells in a pale and yellow ommatidia of Drosophila melanogaster. The top row shows 2 of the 6 outer photoreceptor cells, R1, R6, and the inner R7 and R8 cells. The bottom row shows the different opsins, R1, R3, R4, RH5, and R6 the cells express. Figure from Sharkey et al. 2020. 143. Expression of the opsin RH1 in the photoreceptor cells R1, R6. The genome of Drosophila encodes 7 opsins, 144. 5 of those are expressed in the ommatidia of the eye. The photoreceptor cells R1, R6 express the opsin Rh1, 145, which absorbs maximally blue light around 480 nanometers, 146, 147, 148. However, the R1, R6 cells cover a broader range of the spectrum than an opsin would allow due to a sensitizing pigment 149, 150 that adds to sensitivity maxima in the UV range 355 and 370 nanometers, 148. The R7 cells come in 2 types with yellow and pale rhabdomeres, R7Y and R7P, 151, 152. The pale R7P cells express the opsin Rh3, 153, 154, which maximally absorbs UV light 345 nanometers, 155. The R7P cells are strictly paired with the R8P cells that express Rh5, 154, which maximally absorbs violet light 437 nanometers, 148. The yellow R7Y cells express a blue-absorbing screening pigment 151 and the opsin Rh4, 156, which maximally absorbs UV light 375 nanometers, 155. The R7Y cells are strictly paired with R8Y cells that express Rh6, 157, which maximally absorbs UV light 508 nanometers, 148. In a subset of ommatidia, both R7 and R8 cells express the opsin Rh3, 154. However, these absorption maxima of the opsins were measured in white-eyed flies without screening pigments Rh3 or 6, 155, 148, or from the isolated opsin directly Rh1, 146. Those pigments reduce the light that reaches the opsins depending on the wavelength. Thus, in fully pigmented flies, the effective absorption maxima of opsins differs, and thus also the sensitivity of their photoreceptor cells. With screening pigment, the opsin Rh3 is shortwave shifted from 345 nanometers B to 330 nanometers, and Rh4 from 375 nanometers to 300. Whether screening pigment is present does not make a practical difference for the opsin Rh5 for 135 nanometers and 437 nanometers, while the opsin R6 is long wave shifted by 92 nanometers from 508 nanometers to 600 nanometers, 143. Additionally, of the opsins of the eye, Drosophila has 2 more opsins. The ocelli express the opsin Rh2, 158, 159, which maximally absorbs violet light approximately 420 nanometers, 159, and the opsin Rh7, which maximally absorbs UV light 350 nanometers with an unusually long wavelength tail up to 500 nanometers. The long tail disappears if a lysine at position 90 is replaced by glutamic acid. This mutant then absorbs maximally violet light for 150 nanometers, 160. The opsin Rh7 entrains with cryptochrome, the circadian rhythm of Drosophila, to the day-night cycle in the central pacemaker neurons. 161. Each Drosophila opsin binds the carotenoid chromophore 11-cis-3-hydroxyretinol via lysine. 162. 163. This lysine is conserved in almost all opsins. Only a few opsins have lost it during evolution. 164. Opsins without it are not light sensitive. 165, 166, 167. In particular, the Drosophila opsins Rh1, Rh4, and R7 function not only as photoreceptors, but also as chemoreceptors for istoloic acid. These opsins still have the lysine like other opsins. However, if it is replaced by an arginine in Rh1, then Rh1 loses light sensitivity, but still responds to istoloic acid. Thus, the lysine is not needed for Rh1 to function as chemoreceptor. 166. Phototransductions in vertebrate vision. Visual transduction in invertebrates occurs via a G protein-coupled pathway. However, in vertebrates, the G protein is transducin, while the G protein in invertebrates is GQ-DGQ in Drosophila. When rhodopsin Rh absorbs a photon of light, its chromophore, 11-cis-3-hydroxyretinol, is isomerized to all-trans-3-hydroxyretinol, or undergoes a conformational change into its active form, metarrhodopsin. Metarrhodopsin activates GQ, which in turn activates a phospholipase CPLC known as NORPA. 168 PLC hydrolyzes phosphatidylenosyl 45 bisphosphate PIP2, a phospholipid found in the cell membrane, into soluble inositol triphosphate IP3 and diacylglycerol DAG, which stays in the cell membrane. DAG, a derivative of DAG, or PIP to depletion, cause a calcium-selective ion channel known as transient receptor potential TRP to open, and calcium and sodium flows into the cell. 169 IP3 is thought to bind to IP3 receptors in the subrhabdomeric cisternae, an extension of the endoplasmic reticulum, and cause release of calcium. But this process does not seem to be essential for normal vision. 168 calcium binds to proteins such as calmodulin, CAM, and an eye-specific protein kinase PKC known as INAC. These proteins interact with other proteins and have been shown to be necessary for shutoff of the light response. In addition, proteins called arrestins bind metarrhodopsin and prevent it from activating more GQ. A sodium-calcium exchanger known as CALX pumps the calcium out of the cell. It uses the inward sodium gradient to export calcium at a stoichiometry of 3-nol-1 Ca++. 170 TRP, INAC, and PLC form a signaling complex by binding a scaffolding INAD. INAD contains 5 binding domains called PDZ domain proteins, which specifically bind the C termini of target proteins. Disruption of the complex by mutations in either the PDZ domains or the target proteins reduces the efficiency of signaling. For example, disruption of the interaction between INAC, the protein kinase C, and INAD results in a delay in inactivation of the light response. Unlike vertebrate metarrhodopsin, invertebrate metarrhodopsin can be converted back into rhodopsin by absorbing a photon of orange light, 580 nanometers. About 2/3 of the Drosophila brain is dedicated to visual processing. 171. Although the spatial resolution of their vision is significantly worse than that of humans, their temporal resolution is around 10 times better. Grooming Drosophila are known to exhibit grooming behaviors that are executed in a predictable manner. Drosophila consistently begin a grooming sequence by using their front legs to clean the eyes, then the head and antennae. Using their hind legs, Drosophila proceed to groom their abdomen, and finally the wings and thorax. Throughout this sequence, Drosophila periodically rub their legs together to get rid of excess dust and debris that accumulates during the grooming process. 172 grooming behaviors have been shown to be executed in a suppression hierarchy. This means that grooming behaviors that occur at the beginning of the sequence prevent those that come later in the sequence from occurring simultaneously, as the grooming sequence consists of mutually exclusive behaviors. 173, 174, this hierarchy does not prevent Drosophila from returning to grooming behaviors that have already been accessed in the grooming sequence. 173. The order of grooming behaviors in the suppression hierarchy is thought to be related to the priority of cleaning a specific body part. For example, the eyes and antennae are likely executed early on in the grooming sequence to prevent debris from interfering with the function of D. melanogaster sensory organs. 173, 174. Walking top view of a walking Drosophila, left with legs tracked with diplapkit. 175 right. Like many other hexapod insects, Drosophila typically walk using a tripod gait. 176. This means that 3 of the legs swing together while the other 3 remain stationary, or in stance. Specifically, The middle leg moves in phase with the contralateral front and hind legs. However, variability around the tripod configuration exists along a continuum, meaning that flies do not exhibit distinct transitions between different gaits. 177 at fast walking speeds. The walking configuration is mostly tripod 3 legs and stance, but at slower walking speeds, flies are more likely to have for tetrapod or 5 legs and stance wave. 178, 179 These transitions may help to optimize static stability. 180 Because flies are so small, inertial forces are negligible compared with the elastic forces of their muscles and joints or the viscous forces of the surrounding air. 181 Flight flies fly via straight sequences of movement interspersed by rapid turns called saccades. 182 During these turns, a fly is able to rotate 90 in less than 50 milliseconds. 182 Characteristics of Drosophila flight may be dominated by the viscosity of the air, rather than the inertia of the fly body, but the opposite case with inertia as the dominant force may occur. 182 However, subsequent work showed that while the viscous effects on the insect body during flight may be negligible, the aerodynamic forces on the wings themselves actually cause fruit flies' turns to be damped viscously. 183 Connectomemine article. Drosophila connectomedrascil is one of the few animals, C. elegans being another, where detailed neural circuits, a connectum, are available. A high-level connectum, at the level of brain compartments and interconnecting tracts of neurons, exists for the full fly brain. 184. A version of this is available online. 185 detailed circuit-level connectums exist for the lamina, 186, 187, and a medulla, 188 column, both in the visual system of the fruit fly and the alpha lobe of the mushroom body. 189. In May 2017, a paper published in Bioarxiv presented an electron microscopy image stack of the whole adult female brain at saptic resolution. The volume is available for sparse tracing of selected circuits. 190, 191. Since then, multiple datasets have been collected, including a dense connectum of half the central brain of Drosophila in 2020, 192, 193, and a dense connectum of the entire female adult nerve cord in 2021, 194. Generally, these datasets are acquired by sectioning the tissue, e.g. the brain, into thin sections on order of 10 or hundreds of nanometers. Each section is then imaged using an electron microscope, and these images are stitched and aligned together to create a 3D image volume. The methods used in reconstruction and initial analysis of the such datasets followed. 195. Due to advancements in deep learning, automated methods for image segmentation have made large-scale reconstruction, providing dense reconstructions of all the neurites within the volume. 196. Furthermore, the resolution of electron microscopy illuminates ultrastructural variations between neurons as well as the location of individual sapses, thereby providing a wiring diagram of saptic connectivity between all neurites within the given dataset. In 2023, the complete map of a Drosophila larval brain at the SAPS level and an analysis of its architecture was published. The larval brain consists of 3,016 neurons and 548,000 saptic sites, 197, whereas the adult brain has about 150,000 neurons and 150 million sapses. Misconception: Tris is sometimes referred to as a pest due to its tendency to live in human settlements where fermenting fruit is found. Flies may collect in homes, restaurants, stores, and other locations. 15. The name and behavior of this species of fly have led to the misconception that it is a biological security risk in Australia and elsewhere. While other fruit fly species do pose a risk, D. melanogaster is attracted to fruit that is already rotting, rather than causing fruit to rot. 198, 199. Thanks for watching.